the door. So signal that nobody's out there eating snacks. Please eat more snacks afterwards so that I don't have to bring them home or eat them myself. So it is my pleasure to introduce collaborator and friend, Dr. Ariana Kuhn. Ariana is currently the Curator of Herpetology at the Virginia Museum of Natural History. Ariana received an MS at Villanova University, working on systematics of African lizards. She then saw the light and switched her research program over to snake systematics for her doctoral research, which was conducted at City University of New York and at the American Museum of Natural History. That work focused primarily on Malagasy snake phylogeography and historical demography. She was then briefly a postdoc at University of Lethbridge in Canada before landing her current gig as the Curator of Herpetology at the Virginia Museum of Natural History. Since starting at the Virginia Museum, she's expanded into community diversity of salamanders, and she continues her work on Malagasy squamates as well. And I just realized I did not read our land acknowledgement, but I do want to acknowledge that the Field Museum stands on the lands of Native peoples of this region before I introduce Ariana here today. And so with that, I would like to welcome Dr. Ariana Kuhn.
Okay. Yeah, I'm so excited to be here. This is my first time visiting the Field Museum, and it's such a fantastic institution. There's phenomenal research collections here. And as someone that's just started as our first Curator of Herpetology at the Virginia Museum of Natural History, which is a smaller state institution, there's so many opportunities to learn and support our growth through connections to larger institutions like the Field Museum. So I'm very grateful and excited to be here today. So the questions I'm going to be talking about today are really something that I've always been drawn towards, even since I was a little kid. These questions are all about how organisms exist in their natural environments and how these types of interactions can tell us more about why they're there or why they're not there. And then these questions have certainly evolved since the picture on the left up here. But what I really want to know, these underlying reasons are the same, but the questions really evolved to, can we explain the causal mechanisms that have generated the patterns of biodiversity that we see in our world today? And I answered these, so to simplify this question even further, I want to give an example from where I'm from. So I'm from southeastern PA, and I grew up here catching snakes. So these are some of my local backyard snakes. And if I were to go visit my folks and go out and catch snakes, which they absolutely love when I come home and do this, I might find some milk snakes, I might find some rat snakes, and there might be other species I see, and then there's going to be certain species that are never going to be here no matter how many times I go out and look for them. So I have a lot of curiosity from things like this in my natural environment about how certain organisms got to be there, what external factors like their climate, environment, or intrinsic factors like their physiology or their life histories kind of caused them to originate, caused them to diversify over time, and allows them to be maintained in challenging conditions. And can we use this information to model and predict how these species might fare under changes in their immediate future? So the approach I take to answering these questions is very similar to other people here at the Field Museum. I love to get out in the field. This is where everything starts. So we're packing up our vehicle. In this photo, that vehicle is in Madagascar, but sometimes the vehicle I pack just goes 20 minutes north of where I live now into the field, to the habitats that my study organisms live in. And here I'm collecting field observations, in situ photographs, census and survey data from these organisms, interacting with their natural environments. This is really one of my favorite parts about being a biologist. This is the hook for me when I first started getting into some of these things. But this isn't the only data that I need to answer those questions I just talked about at the beginning here. Another important part of my research program involves museum-based collections work. That's why I'm here. That's where a big part of my research program is drawing information from. Things collected by myself in the field or those things collected and existing in museums for a very long time. And these trips are also really important because of other pieces of data that I bring back. In this case, these are tissues from organisms that I can extract DNA from and answer a ton of different questions. And depending how those tissues are preserved, we can get everything to capture the history of the organisms, what genes are being expressed at the cellular level, and well beyond that. And we're even able to extract DNA from really old specimens now. And that's like a lot of Sarah Ruan's work that you might be familiar with. But this is also just giving us this holistic picture of what happened in the past. And it also gives us a tool to start to predict how that might shift in the future. And you may have already guessed from the organisms and the fact that I'm here with the herpetology department for this seminar, the organisms that I study are herps. And I love studying reptiles and amphibians because questions about biodiversity across these huge gradients are great with a group that's almost everywhere. So this is just showing the distribution of amphibians globally, but what you can see here, and this pattern holds true for reptiles as well, is they're almost everywhere. And they're not just your typical ectotherms that are really restricted by this life history trait, but they can be found across these diverse ecoregions. You can have frogs that are freezing and thawing. You can have things like a frog that you expect to need a moist environment, and yet these desert rain frogs are existing in these really, really arid environments. They're also found across a variety of habitats. You can have snakes that are entirely pelagic. You can have frogs that are nearly fossorial throughout their lifetime, and you can have salamanders that are arboreal. So places we really might not have expected to see these animals in their typical understanding, they're still there. And then lastly, they have really cool and diverse ecologies. And these are everything from the things they eat, like this African egg-eating snake that's got no teeth and is an obligate egg feeder, to things like this slug-eating snake that has specializations in their behavior and morphology for eating mollusks. And so I investigate herpetological patterns of biodiversity across hierarchical scales. So this can be everything from population genetics, where I'm focusing on a single species at really strong local resolution, as well as taking a lot of these different populations and asking about comparative phylogeography on a more regional scale. And then lastly, scaling this up even more to ask questions about the entire communities as they're distributed across large regional spaces. And what I want to kind of just highlight about studying these different levels of biodiversity is what I've found, and what I'm really interested in exploring more, is that there's feedback among all of these levels, despite the fact that historically, sometimes the communication and the types of data you generate has not always been feasible. From a modeling perspective or from a data generation perspective, but these patterns and the way that they're shared is really interesting to me. And finalizing that with saying that taking things like genes and combining them with traits and the environments that organisms exist in across these different scales of biodiversity gives us a tool to understand the past, which is a really important piece of predicting the future. So for my first example of how I explore these origins of biodiversity, I'm going to take us out of where my museum is located, in Virginia Museum of Natural History in Southwest Virginia, zoom us into the southern hemisphere across the Mozambique Channel to the island of Madagascar, and what you can already take from this map here that's showing us the number of endemic species by terrestrial ecoregion is that endemism is incredibly high on this island. It's been isolated from other continental land masses for nearly 80 million years. And as a result, there's just this incredible assemblage of high biodiversity with extreme endemism on the island. And these questions I outlined at the beginning of my talk, they're great questions to ask anywhere. But when you ask them on this island, that's essentially a closed system, teeming with diversity and these massive in situ radiations across a restricted number of families, we can really start to get into the individualistic processes that have generated these patterns that have driven this high biological diversity on this island, and what is maintaining this biological diversity through time. So in addition to this long-term isolation, Madagascar also has a wide variety of ecoregions and habitat types across its space. So it's about the size of California, and yet it's almost like a continent in the way that it operates with all these different ecoregions. So perhaps those that it's best known for are going to be these lowland and highland eastern humid rainforests, where you might see something like one of my favorite snakes on earth, this Liophidium petoni, just because it's so stunning. This is a real species that exists there with these colors. But there's a variety of other ecoregions on Madagascar. So there's these high elevation central plateau area that has a mosaic of dry deciduous forest pockets. And that's somewhere that you might see this widespread Rickettsia chameleon. They're found all over the place there. But then there's other habitats that are going to be like this karst limestone formation that's found in a few places on the island, where you have things like this Paradura that's highly specialized for that sort of karst limestone formations lifestyle. And what I wanted to know about Madagascar and these diverse herp assemblages that are found on the island was if discrete but major events in the history of Madagascar could be invoked to explain these patterns that we see about pockets of endemism and shifts in biodiversity across this space. And I focused on answering these questions using reptiles and amphibians as my focal system, because as I told you before, one, I really like them. So that worked out incredibly well. But also this is one of the largest vertebrate groups on the island. So it's taking up a lot of that vertebrate terrestrial mass. And so it's a really great group to study something like a pattern of biodiversity when there's so many organisms in this assemblage. In addition to all these reasons, like herps are really fun to catch, they're really fun to find in the field, and some of the coolest animals, no matter what group of organisms you work on, are found here on the island of Madagascar. Leaf-tailed geckos, really adorable chameleons. Sarah can agree with me on some of that, I think. So I mentioned I was interested in these major changes that are responsible for major ecoregion level change, shaping these patterns of biodiversity. And one of these major changes that we know has taken place in the history of everywhere is climate change. And globally, it was very unstable throughout the Quaternary, so the last 2.6 million years of our past. And to show this graph, just to illustrate it, we have time progressing and we have climate shifting through periods of things maybe being very warm and very wet to shifting to maybe unfavorable conditions. Things are maybe becoming quite dry and they're becoming quite warm. And we have to imagine that in response to these shifts in climate, vegetation is also going to shift, and the organisms that rely on that vegetation as their primary habitat may shift in response as well. So to illustrate this, you can see as things are becoming maybe warmer and wetter and their habitat is expanding, the organism is responding by increasing its population size over this space. And then when things become somewhat unfavorable, maybe that population size in response is shrinking down into refugial pockets throughout the landscape. And sometimes these events are so strong, they could result in a pattern like extinction, or organisms can expand over a large space such that they start to actually adapt locally and form new species. So this is really a tool for generating both species diversity, but also patterns of extinction on the island, potentially. And just like the rest of the globe, we know that in Madagascar, climate has not been historically stable. So over the last 10 million years, things were potentially a lot drier and a lot less warm than they are now, as well as things like precipitation, things could have been a lot drier. So we know that climate was not stable. And what we wanted to know was there's an expectation then that if these events were very strong and had a huge impact on a lot of organisms on the island, such that many things either shrunk in size or expanded in size, this would be an important event that could help us understand large patterns of biodiversity on the island and not just the response of a single organism or a single population. So this question kind of boils down to, were expansion events where populations are all becoming larger in response to historical climate kind of temporally congruent with these events, and were they happening synchronously, meaning that everybody was kind of doing it around the same time and not all over the place? And the approach I took to kind of answering this question was using genomic data, but the genomic data was never in isolation. As I mentioned in the beginning of this talk, I'm always kind of coupling that with trait data from organisms, and then I'm coupling it with data from their local environment. So through environmental layers to get this holistic view of what were all the factors that were driving responses if they weren't all the same in the communities. So as I mentioned before, it's not just the genomics of the organisms and their response, it's the ecoregions that they're distributed in. So what we do know about Madagascar is each one of these major ecoregions I've highlighted here had an independent biogeographic history with sometimes local endemism within each of these ecoregions, but some things were distributed across all of them. So the first question I had was, how did this play a role in responses of organisms? In addition to that, we know that not all these species I've been showing in these slides are the same kind of animal. They have a lot of different things going on from the life history perspective. So just as an example, yes, these are both geckos and they're going to share basic gecko traits between both of them. However, this Paradura is terrestrial and you're going to find these out more during the day. And then you're going to have something like a Europlatus that's going to be nocturnal, and these are completely arboreal. So these animals aren't actually operating in the same realm, although they do share some traits. And then kind of moving on from there, we have organisms that are as different as a gecko and a frog, meaning that they could have had different biogeographic origins. When did they colonize the island? When did they diversify? So some differences here, even though both of these organisms do share the fact that they're in the same terrestrial environment. So my questions kind of asked whether shared traits, such as both of you being a frog and having just typical amphibian traits, were driving responses to be more similar, or if it's things that the ecoregion you're in is much more important in your response to something like a shared trait of both being an amphibian. And the way that I addressed these hypotheses was using a model-based simulation approach and using machine learning inference to kind of estimate different parameters that relate to answering this question. So just to outline this model, you can imagine each one of these little strings here being an independent population trajectory. And when that pipe gets larger, the population is expanding, and that dotted line is kind of identifying everyone expanding at the same time. So this is my synchronous hypothesis. And if I actually find that time links up to the same time that climatic things were shifting on Madagascar, this addresses my hypothesis that maybe historical climate was a really important driver, regardless of organismal traits, to those population level patterns. However, my alternate hypothesis to this is really that things could be highly asynchronous, meaning that every one of these types, every one of these independent populations is changing in size at a very different time in the organism's history, such that there's a bunch of different explanations for why we might see this pattern. Could be that a prey item expands and then that population that preys on that item expands much later. So there could be a delay. It could be that you have an independent biogeographic history, like you weren't there when the change was actually happening. So there's no record of this in your history. And a variety of other explanations as to why things might be a little bit asynchronous. And what I will tell you, I'm going to jump right into the results, is that across all of Madagascar and across all these really incredible and diverse organisms, things were highly asynchronous. A lot of populations were expanding at very different time periods across all of these ecoregions. However, one thing that was really interesting that I did find was that when you shifted over to the eastern humid rainforest, we found that the responses of these organisms, no matter what organism you were very synchronous. So when we kind of ruled everything out and pared it down to this humid environment, a lot of things were expanding, and that expansion time was about 100,000 years ago, that co-expansion time just after the last interglacial. And this highlights the importance of these humid regions as they have acted as these like refugial zones for forest adapted taxa. And these humid corridors connected suitable habitat across elevational gradients during unfavorable periods. And so they played a really huge role in conserving genetic diversity for species level endemism across the entire island. So not just the humid rainforest, but kind of everything ended up expanding back out of these refugial zones in the eastern humid rainforest. And this is important because when we just examine a single section of these eastern humid rainforests, what we already know is only about 8% of intact forest is left on Madagascar. So these are just true estimates from 2000 and 2012 of actual forest cover from this region. And this holds true for the rest of that eastern humid section. But these projections based on current estimates show that we're going to lose even more. And this is really important because this time around, we no longer have the buffer for future climate change that these humid corridors provided in the past for a lot of species. And there's a huge amount of the diversity of Madagascar is distributed across this really unique ecosystem. So just kind of identifying the fact from this first study that I'm sharing with you that these eastern humid corridors are really important, but by understanding the past history of these organisms, we can understand just how important and what role they play in conserving genetic diversity. And that's something that we're going to be looking into on a more localized scale in the future with other species. So my study looking at all reptiles and amphibians kind of highlighted the importance of species-specific differences and demographic response, but it also showed that extrinsic factors, so things like which organisms we were looking at and what life history traits they had, kind of played a role in the response of different organisms. But to really dig into these extrinsic factors themselves, I needed to work with a group that we understood the tempo and the timing of their colonization on the island. And a really great group for that is a group that I feel very strongly about, which are the Malagasy gem snakes. And I think Sarah does too. So this is kind of a fascinating radiation on the island. There's a lot of diversity in this group. They're occupying all these different really interesting niche spaces. And yet we really understand the colonization of the island by this radiation and the timing of different speciation events. So they were a perfect group to ask these questions. And a lot of work went into collecting this level of sampling for snakes. It's really difficult to find a lot of snakes in the field. So everyone that was involved in this project contributed a lot to finding these animals. And it would not have been possible without this fantastic team of people that were all helping. And also it was really fun to have an excuse to go back to the field to check out these animals in person and see what was going on. And so this brings me down to my next question about these responses across these different snake assemblages, whether or not expansions and also bottleneck events where populations are contracting were related to historical climate and range restriction. So I was able to ask this question about endemism as well. And I wanted to know if these interactions between population dynamics actually told me anything about diversification of the group. So linking across these different hierarchical levels of biodiversity. So because we knew a lot more about these snakes from the work that Sarah actually had done well before I got there, we understood the timing, we understood the diversification, and we understood the distribution for a lot of them. So I was able to also ask another question that was very interesting to me that had to do with whether or not being incredibly range limited had anything to do with the way that organisms could respond to these historical events. So we had a lot of snakes in this assemblage I was looking at that are found everywhere. So you can look behind me in this photo. This is really degraded habitat. You're going to be able to find these giant Malagasy hognose snakes that are not related to the hognose snakes that we have here in North America at all. You can find them in a lot of different habitat types. They're somewhat of a generalist. And yet we have other species that are going to be found in a more restricted area closer to the eastern humid rainforest. So even though they could be abundant in this habitat type, they're not going to be found in the more dry deciduous parts of the island. So they're a little bit more of a specialist in that way. So I could ask this question within this group, now that I was controlling for this idea of colonization history and diversification history, which is, does it mean anything to be more endemic? And was that going to dictate the type of response that you had to these historical climate events? And so we were able to take this large group, this large amassing of RAD-seq data to answer this question. So a combination again of traits, genome-wide sampling data, and lots of different populations of gem snakes from the island. And we were able to estimate their expansion histories to identify some as having bottlenecks, some of having expansion events. And what I can tell you right away is that endemism didn't appear to have to do anything with their responses. So when we put this into a predictive modeling framework, whether you were here or you had a very large range, did not give any predictions as to whether or not you were going to expand or have contracted in response to historical climate change amongst the 33 populations that I looked at. However, when we looked at synchronicity in expansion across this data set, we found high levels of shared responses across all of these gem snakes around 120,000 years ago. And this is really significant because Sarah and other collaborators, including myself, had previously reconstructed the entire evolutionary history of this group using a ton of genomic data and a ton of samples. And what they found is that speciation rates were relatively consistent throughout the Miocene. When we look here and zoom in on the Pleistocene, there was a massive drop-off in these rates of speciation. So over the last several 100,000 years, right around the same time that I was actually seeing a lot of population expansion events for this group, that typically can result in things like colonizing new habitat and forming new species. It appears when we're looking at the diversification history of the entire group, that the Pleistocene was not a good time for speciation. And in this study, Dr. Ruan and others identified 38 new species, and almost 90% of these new species appear to have originated in the Pleistocene. The same time that we were actually seeing this drop off in speciation rates. So these results, coupled with my findings here, suggest that not only was most of the standing extant diversity in this group generated during the Pleistocene, but that Pleistocene shifts in climate change was probably still a really important factor in driving the origin of these new species. And when we see these rates dropping off, it's probably due to other factors such as saturation of niche space through interspecific competition. When we looked at bottleneck events, so again, that one of those factors that I talked about in the beginning of this talk that can result in populations not just becoming smaller, but maybe even going extinct. We found a lot of synchronicity in this as well. So across all these gem snakes, the proportion of co-contracting populations, so populations all getting small at the same time, was very high as well. And this happened around 45,000 years ago. And this is really important to me, or hopefully others, because this all happened before the onset of the Holocene and prior to the period of the last glacial maximum. This is important because the loss of endemic megafauna on Madagascar, such as these enormous elephant birds, the giant lemurs, and so forth, this is really still debated in the literature. And the arrival of humans has been invoked as a potential cause of the loss of this megafauna, but it's not entirely certain what was the cause. However, what we do know is that these species all experienced several periods of extremely warm and arid conditions, so extremely unfavorable conditions for these species over the past 8,000 years. What we also know is that human arrival on islands and continents has almost always led to a dramatic loss in biodiversity. But some evidence on Madagascar suggests that not only was this due to this period of climatic stress, coupled with increased human activity, but also a current kind of prominent drying period. So sort of like a smoking gun of all of these things coming together, not just humans, not just historical climate change, but vulnerable populations that experience bottlenecks that then were faced with something that they didn't have the same genetic diversity to deal with. So my study here kind of underscores the importance of looking at past history to potentially identify vulnerable populations and have a better understanding of how they might respond to kind of very difficult situations in their near future that might require that same genetic diversity. So I've shown you that these demographic events, especially at the community or assemblage level, can have populations expanding, we can have populations contracting, and this can happen multiple times throughout that species level history. But it can also result in these other really important factors such as extinction and speciation events. And these two patterns themselves can scale up to the assemblage level across entire regional spaces to generate these really important patterns that I've said I was interested in from slide 1, which are things like centers of endemism. Why across different groups do we see pockets that seem to be shared for where everything is endemic, and other places where it's very different for different groups of species, as well as patterns like species richness. So if we look down sometimes from the population level histories, how those dynamics then influence patterns like speciation and extinction, and scale that up to look at diversification across entire groups and entire regional spaces, we can get a really awesome picture of where these patterns of biodiversity that I think are so incredibly interesting really come from and what's driving those patterns. And we know that these historical events have left a signature on the genomes of organisms. And we also know that in present day, we're experiencing events not completely unrelated to something like historical climate change, things like habitat loss, things like disease, things like invasive species. And sometimes these events themselves also leave a signature on the genomes of organisms that we can detect when we generate genomic data from tissues that we collect in the field. And so the second part of this talk that I want to give an example of my research program kind of leverages genomic data coupled with traits, coupled with environmental data to ask this question of, can we detect the signature of these types of impacts from genomic data of organisms? And can that be used as a really great tool to inform conservation action? And the more and more we're experiencing this loss of biodiversity in present day, and it's going to increase in our future, genomic insights are becoming increasingly an important part of the recovery strategy for a lot of different species. And so during my postdoctoral work in the University of Lethbridge in Southwest Alberta, I started becoming involved in projects that were using these tools to understand how parks could better conserve and remediate changes that were happening to native amphibian populations. So these are the long-toed salamanders. Alberta only has 2 salamanders, which was such a crazy shift to then come to Virginia where there's 1 billion species. So a very interesting and different environment to be a herpetologist in. They don't have nearly enough herps there in terms of diversity. But what I wanted to use these genomic tools for was to understand how historic anthropogenic events impacted populations, just like I was looking at the impact of historical climate on populations, and how maybe genomic data from things like museum collections sampled temporally, so in the past and in present day, can be useful to applied conservation initiatives. So as I mentioned, this work was with the long-tailed salamander. This is one of our Pacific Northwest distribution mole salamanders, so not too distantly related to, say, something like a spotted salamander that you might find here. And not only are they distributed in Canada, but they're also throughout all the Pacific Northwest in the United States. So we know there's a little bit of phylogeographic structure in this group, and we know that they're of concern in Alberta, but they were not yet at extreme risk. And that gave us an opportunity to actually explore these questions of how can we use genomic data in a group that's not completely on the brink of extinction, and therefore we have very few options for how we can utilize that genomic data no matter what we find. And so in Alberta, just like many other places, most of the water bodies that could support fish and be stocked with fish have been stocked with fish. So every one of these green dots is a place that we know some type of lake or water body has been stocked with non-native fish. And what this has caused is local extirpation of salamanders, especially in the national parks where it's being documented and studied. So fish get there and the long-toed salamanders are gone. They're eating all the larvae and they're no longer found in that habitat. And Waterton Lakes National Park, which is a national park in Alberta. So if you think of where you know that Glacier National Park in Montana might be, this is sort of like just like a little hat on top of it as you move into Canada. So very similar habitats. So this park is the first park that was like, we're very interested in reintroducing these salamanders, but because they had interest in choosing from a lot of different source populations, they actually linked up with us at the University of Lethbridge to say, maybe there's another tool we can use to inform this reconstruction so it's actually successful and not just a black box of what we're choosing and where we're putting it. And so this was a fantastic opportunity to kind of create this workflow and these targets of answering questions that draw on stuff that I'm really interested in, which is how does the organism's biogeographic history and species history actually inform this? And how can we use these tools to make better predictions and have more successful reintroduction events, as we know it's going to be something that we're going to have to do a lot more in the future potentially. So how far can we go outside of the known range of where we want to reintroduce this thing and still actually be recovering the same lineage? And you might think to yourself, there's no way that a reintroduction could be bringing in the wrong species. And I'm going to tell you that there's a lot of reintroductions that happen that might bring in the wrong lineage by not investigating their phylogeographic history beforehand. And we also wanted to know about the source health. Not every population is going to be genetically as intact as others, just due to things like habitat fragmentation. So we wanted to investigate all the sources and pick the one that was the healthiest. And we needed to know whether or not source populations could be locally adapted to different habitat types. And therefore, you could take something that looks like the same salamander that's really adapted to high elevation conditions and then put it at low elevation. And therefore, it's no longer in a habitat that's suitable for local adaptation. So what I will tell you is that each one of these colored blocks that we found when we investigated the species history throughout that Pacific Northwest distribution is there's a lot of very different and very evolutionarily distinct units within the long-toed salamander complex. So everywhere you see a different color is something that's very, very distinct. So up to 6 different lineages are actually found here. And the most important thing I want to point out is right where the green and the yellow cut across one another in Southwest Alberta are 2 different species. So if you take your national park where we're working and you bump up to Banff National Park or Jasper National Park, just north of there, you are no longer sampling the same lineage. This has a completely unique evolutionary history and would not be the right population to reintroduce into Waterton Lakes National Park. Secondly, we investigated source health. And a lot of these salamanders are distributed at incredibly high elevations and others are found at lowland population elevations. And what we found is as you move down to higher or move up to higher elevations, we see a loss of genetic diversity, probably because of isolation. This is something that we're still looking into because this data is relatively new. But what we do know is we might need to source from a high elevation population to have it be an adaptive and habitat match. However, a lot of these high elevation populations are already looking unsuitable to use as sources. Additionally, Waterton Lakes National Park experienced this massive, massive fire. And even though fires are a part of this park's history, this one was so large that huge portions of the park were burned. And looking into this, we wanted to ask, did this burn zone have any impact on finding healthy source populations? For example, do we really want to source populations that were within this burn zone if they're no longer genetically healthy? And what we already found is everywhere you see one of these little tiny flame emojis is a population where sampling historically and sampling in present day has showed us a massive drop off or a massive increase in things like inbreeding coefficients or a massive loss of patterns like allelic richness. So some of these burn zone populations, although the original habitat match would have been great for some of our reintroduction sites, they're no longer going to be the best populations that we might want to source from, or we might need to source from a bunch of different populations. And last, the adaptive fit. So we're part of a larger initiative to biobank species of interest in BC and in Alberta. And so this is a great opportunity to build these long-term genetic resources. And in the case of the long-toed salamander, whole genome resources. And this allows us to ask this question of, if we do want to put things into a high elevation habitat, is it locally adapted, or are all these shifts in high and low elevation salamanders actually quite plastic? So these are 2 individuals that were caught one day apart at very different elevations. And you can see there's already a huge shift in their body sizes. So there's really big developmental differences. But we don't know if these are adaptive or if this is something that's just plastic across this group. And these genomic resources for this biobanking project are going to give us an opportunity to answer that question. So this is an example of these low elevation sites. Some of them are just off the side of the road. So even though I told you all this great stuff about genomics, there's a feasibility and financial aspect to working in applied conservation genomics that needs to be taken in consideration. This would be a great place for Parks Canada to source their populations. It's on the side of the road. Other places like this that were really stunning, and I had a great time sampling here, took us 2 whole days just to get 6 salamanders and 2 whole days just to hike up to the site. So unless Parks Canada wants to get a bunch of helicopters and go up here, even though it might be the perfect fit for source population, it's never financially or feasibly going to work for this project. And so those types of things are being taken into consideration. So this is a great opportunity to understand, like, how can we take the kind of tools that we as museum scientists are using and make them tools that applied scientists can actually use and take into consideration. So as we move forward, we're going to add a lot more sites now that we know how far outside the park boundaries we can go. We're going to incorporate stuff like species distribution models with presence and absence sampling to know if the source sites are the same match as where the reintroduction sites are, and not just the genetics of the organisms themselves, but also their environments, and add in these whole genome resources. And lastly, develop these post-monitoring programs. Because if we never go back and find out if it worked, we're never going to know if all the work that we did was actually helpful to inform future reintroductions of other species. And this type of work, now that I'm in Virginia, continues in this highly diverse salamander spot where we're able to bring in, this is all very relatively new, but we're able to understand, get our thermal guns and learn about thermal preference across all plethodontic salamanders in the region, and take them through these little thermal mazes where the salamanders do nothing that you want them to. So we can learn about how thermal preference across this group of organisms interacts with things like disease monitoring and future conservation initiatives in the area. And lastly, this is an initiative and my final question that I think I probably share with everyone in this room, which is how do we leverage museum resources to train lifelong learners and the next generation of wildlife ambassadors? And working with herps has been a really fun opportunity to kind of share this passion I have for this group of organisms with everybody that I meet that isn't running away from the snakes that I'm holding. So, and if not, then there's always salamanders for people that feel that way. So I want to share a little bit about my museum, because I don't know if everyone in this room would necessarily know about the Virginia Museum of Natural History. It's relatively young comparatively, so founded in 1984, accredited only in 1994. We've got 9 exhibits, 2 are rotating. And although this is not a large museum, it fills a really important gap for kind of rural communities and schools to have a really cool resource in Southwest Virginia. And, you know, we also do a lot of research and maintain pretty interesting collections that have a cool regional focus that fills, again, an important gap in that Virginia is situated somewhere where we capture in the Appalachian region a lot of northeastern diversity of organisms. And then we also have the cool opportunity to capture the northern extent of southern distributed lineages out toward our sandhill regions and the coast. And again, an opportunity to go across these cool elevational gradients all the way from the Appalachian Mountains down to those coastal plains. So again, our focus is very regional, but there's some cool strengths in the collection across invertebrate zoology, paleontology, archaeology, and you know, I've been there since October, so I'm learning a lot about these different collection spaces. And lastly, I just want to highlight the fact that historically there's been a collaboration between the Field Museum and the Virginia Museum of Natural History. So we have one of the best millipede collections of any museum. It's in the probably top 10. This has been a huge focus of historical curators that also collaborated with curators from here on this. And these collaborations are still continuing today. So I was informed by our myriapodologists that these are all representatives of this group of cool shivelback millipedes. Then they're working on a larger vision with scientists from the Field Museum as well to understand this group. And you know, within our collections, although they're not the largest in the world, we do have a lot of cool stuff. We have over 500 types and over 200 undescribed genera of millipedes, which is really kind of fascinating to me. And I'm happy to be a part of this institution. And again, where we're located geographically highlights a lot of the regional diversity that I'm really interested in. So we can have everything from cave habitat salamanders to sandhill salamanders. So capturing, again, a lot of really cool biodiversity that fills important gaps. I'm always excited to have new collaborations and maintain the ones that I have by providing samples from this region if it's of interest. And finally, as I mentioned before, we are somewhat geographically isolated. So trips like this to talk to other scientists as such a renowned institution is really, really cool for me. And I'll bring back all this information to my museum, and they'll be so appreciative of this connection that we can have. But again, we fill this really important gap in having a lot of these festivals and outreach events that the entire community of Southwest Virginia and beyond comes to, and opportunities for students to learn and execute research projects within the space of a real collection. So I'm excited to continue this dialogue to talk more about how those types of tools are being used at the Field Museum and how I might be able to bring some of that back and make it fit into the programming at the Virginia Museum of Natural History. And I'm just going to end with saying that this research journey has brought me to some of the most beautiful places that I've ever been. And I'm excited to be here. But that last question being, to me, all these places are beautiful, but if I can't kind of train that next generation of lifelong learners, then all of that is kind of lost to the next generation. And so I'm happy to be a part of this scientific community and be able to share these ideas with you here today. And with that, I just want to thank everybody that was a huge help in making any of these projects happen, and also say thank you to some of my ongoing collaborations and historical collaborations with stuff happening in Madagascar. And with that, I can take some questions.
Thank you so much for your talk, Ariana. I have a bunch of questions, but I'll just limit it to one for now. So I was wondering about the gem snake synchronicity. So you mentioned that when you were looking more broadly at like all different groups, that there was this asynchronicity, but then you saw synchronicity within the gem snakes. So do you see synchronicity among snakes in general, or specifically only gem snake species are synchronous with each other, but not synchronous with other taxa? And if the latter, what is it about the biology of this particular group that gives us that pattern?
Yeah, that's a great question. So what I will say is on the island, there is also boids, and I wasn't able to include any of these in the data sets just because of the data that existed. It would have been awesome to have them in there. But so there's 4 large boids on the island, and they were not included in any of these data sets. So I don't know how synchronous they would be with respect to everything else, but they do have a different colonization history than the gem snakes. So that might play into it as well. There were some gem snakes included in that large squamate one, or a squamate and amphibian project, although some of them were not all represented in that. So the gem snake sampling was kind of an independent data set. And although there was some overlap in species, and we did see the same population level responses, there were not as many gem snakes in that total data set to say if that pattern was holding true there. However, what I think would play a huge role in this is that idea that they colonized the island and diversified, and the way that niche space was filled over a discrete time period would have been within that realm of being able to respond to these events. And some of the groups that would have arrived or had very highly endemic distributions in the eastern humid rainforest, that might have been one of the reasons that we saw a lot of that asynchronicity in the data set. In addition to that, I would say that for the gem snakes, a lot of these populations, I had great representation throughout the western dry deciduous region or the northern region of Madagascar. And I didn't have as many that were highly endemic to that eastern humid rainforest. So that's definitely something like I 100% want to look into this further. What's cool is there's whole genome resources now for a bunch of these snakes. And that's an opportunity to really look at these shifts at higher resolution that I think might answer why that's happening a little bit better.
I will ask more questions.
One more question was about that map that you showed with of North America with salamander richness. And there's that big red streak that I guess is the Appalachian Mountains.
Yep,
maybe?
Pretty much.
So I guess I'm just surprised by that because you don't see other peaks where there's other mountain ranges. So can you tell us about salamander richness in that region?
Yeah, I'm so happy you asked about this because it took me a really long time to generate this map. So, and I should explain it further. Everywhere you're seeing a dot is actually a community sampled. So at each one of these sites, it's everything, what's in that community. And then that's interpolated across this space to generate this pattern. And it's really cool because if you take a similar data set with the same sites and do it for frogs, we see a totally different pattern. And then I also looked at this from the perspective of phylogenetic species richness to ask about how that is playing into this. And again, we see really different distributions for frogs and salamanders. And so yes, that region, I mean, where I am located is in the hotspot of salamander diversity globally. It's really, really cool. The only other place that has a comparable level of salamander diversity is going to be down in South America for Boletoglossan salamanders. And so this region, what kind of happens in Appalachia is you just get a lot of micro habitat niche partitioning, which provides a lot of opportunities for speciation. And understanding this pattern better is one of the main goals I had for coming to a place where I could get all these samples. So it's like an ongoing question for me that I'm really interested in. You know, there's been a lot of studies that looked at why this pattern holds true. And yes, there's a lot of microhabitat diversity. There's been a lot of shifts in this region over time that helps drive that, similar to those patterns of climatic change. But this is something that me and collaborators are like, we got to figure this out because it's a really, really cool question to ask about, like, why is it that North America holds the trophy for salamander diversity right in the eastern region across the Appalachian Mountains. So it's really cool. And there's a lot of work that's focusing on how they're partitioning their niche. And I want to look at that question, taking that knowledge and scale it up to a larger geographic space, which has been challenging because they're describing like 10 new species of salamander every year. So trying to like keep up with it. But that's what's really cool. And I love in Virginia is there's still a lot of new species that are being described every year in that region because of this diversity from millipedes, isopods, salamanders. It's really fun. But yeah, that's pretty much what I'm interested in there.
So in Madagascar, you had a synchronous expansion about 120,000 years ago during the last interglacial. And then you had a contraction 45,000 years ago, which is after that interglacial, but not quite at the peak of the last glaciation. Now, those, of course, are primarily northern hemisphere events. Do you think that the glacial and interglacial cycles are affecting things in Madagascar? And how sure you are of that 45,000 year date? Like, what your error bars on that?
Yeah, I didn't show the error bar.
Because it's you know, that's again after the peak interglacial, but it's 20,000 years before the peak of the glacial. So I'm just
totally,
you know, what the relation between glacial cycles and
absolutely. Yeah. So that is a really great question. What I will say is the way that the confidence intervals do bound, the approach that we use and the fact that we had RAG-seq data and a lot of populations. So what increases the resolution in the precision of those estimates is how many populations we're able to include, which is always hard for finding stuff across the Madagascar that's actually population level panmictic sampling. And then the other thing that increases resolution is the depth of genomic data. So if say you're using mitochondrial data, but you had, I don't know, 100 populations, you could really have a lot of resolution there. If you are going to only have 30, then you better have sampling from across the genome. So those confidence intervals, we use this approach where we were kind of, we were simulating a ton of data, modeling that RAG-seq data set, that's my actual empirical data, and using kind of a machine learning inference approach to generate those estimates. And it was actually those confidence intervals are not that wide, such that I can be pretty confident that that 45,000 point estimate is not overlapping with the same period as that population expansion event. Like they're definitely independent. I always wish that the confidence intervals were tighter, but I was actually pretty confident in those estimates. Now, what the patterns on Madagascar has kind of been my question forever. So what I do know is a lot of other studies have focused on one or a few populations on the island, or even for bats, several populations, and have found the signature of change throughout the quaternary being tightly linked to population level responses. But to my knowledge, there's never been any studies that look at these responses using genomic data across the entire island at high resolution with lots of populations. And so I've always wanted to know, we don't have great fossil evidence from Madagascar that tells me about what was happening in the Pleistocene. That's something that we're missing. And so kind of taking a biotic approach to understand the climatic histories is one thing that I'm really interested in. We don't have great resolution, and we don't know how strong or exactly how these shifts that were happening in a way that we really understand in North America or in the Northern Hemisphere were happening in Madagascar. But it is something that I want to know more about. And I can't make them find fossils faster. So this is the approach that I took to answering that question.
We do have a question from the chat.
Oh, fun.
Since you work in a rural area, how do people around you perceive your work? I know there is a huge difference between how rural and urban populations perceive snakes in general.
Yeah. What I would say is the difference I noticed the most, so I did, as Sarah mentioned, I did my PhD at the American Museum of Natural History, so I was in the city. And there, if you take out any kind of snake, everyone's kind of like, whoa, maybe they've never seen one before in person. If I take out a snake or bring up a snake in Southwest Virginia, everyone has had a snake steal an egg from their farm. They're like, oh yeah, this usual suspect is kind of showing up. So it's something that they're familiar with, but maybe what I've found is having more information about that animal that you still see all the time is really, really helpful. And people are pretty surprised and very interested. Like, I'm actually surprised how interested people are in hearing more about these organisms that they do encounter quite frequently. And people are pretty enthusiastic about like contributing and helping to the collections at the museum in that way. And so that has been really surprising and exciting. That being said, because these things are stealing everybody's chicken eggs and getting in the muck there. So someone told me that one climbed up on their counter and ate a bunch of chicken eggs. I don't know about that, but yeah, people kill them a lot, which is a reason why I feel that the work that we're doing for helping people understand something, you know, we've got a lot of copperheads. We have a huge, high density of copperheads in the region. Just having people learn how to identify them and feel more confident in their environments is something that I feel really strongly about doing. And people have been really receptive and really excited about hearing more about that. So that's kind of how I think the work is perceived as in that way.
Okay, we do have another question. Does the richness in salamander speciation relate in any way to that of their predators?
That's really interesting. So there's a lot of different salamanders in this region. We have everything from, you know, these Eurycia that I'm showing here, which are very typical in size. We've got cryptobranchids, so we have giant hellbenders in the region, and these are going to be aquatic salamanders. So we have stream salamanders, we have terrestrial salamanders. We are going to have things that are entirely aquatic, so Necturus or a mud puppy that's never coming out of the water, and they're quite large. We are going to have cool salamanders that have reduced limbs or only their front limbs, so things like sirens. That are also entirely aquatic. So we have a lot of different groups of salamanders, and therefore a lot of different types of predators that are interacting with these salamanders. And when I was saying things like fish extirpating them from their habitat, that's typically fish are predating on things at the egg or larval stage. Even if you're a tiny salamander, you could be looking at a water bug and being in trouble. So there's a lot of different predators of salamanders, from birds to snakes to giant like bullfrogs. And so I have not looked into patterns of predators, but as I mentioned, this pattern that we see here in the eastern United States, the eastern Nearctic for salamanders does not hold true for something like frogs. And I don't believe that it holds true for things like mammals. But as far as other groups of organisms that predate these animals, I don't know about that. But what I have found, because I hang out with the myriapodologists at our museum a lot, is that a lot of the phylogeographic breaks and distributions of salamanders, especially plethodontid salamanders, are very close to that of the breaks they see in millipedes. So biogeographically, this does suggest, and they occupy somewhat similar habitat types. I love it because when we go out looking for millipedes, we always find salamanders. So it's really useful for me. But what that kind of means is there's some patterns that have been so strong in the history of this region that they're not just affecting millipedes, they're also affecting salamanders. So organisms in the same habitat have, and there's some similarities in things that are happening with ants and the salamanders that are eating the ants. So it's something that we've talked about looking into more because I think it's really interesting. But predators, I'm not aware of that pattern necessarily in this region.
All right, if we don't have any more questions from the chat or in person, I want to thank Arianna for giving such a lovely talk to us today. If anybody didn't get a chance to talk to her who wants to, she will be around all week. So reach out to me if you want to have a chat with her or anything, either tomorrow or Friday. And with that, I'm going to say that you are free to leave the stage and I'm going to take you to lunch.
Okay, awesome.